So we pick up from whatever the last day was, and we do our first disciplinary leap as promised at the very beginning of the course. The entire function of the first half of the course is to leap from one bucket to another, such that just as soon as you feel like you are getting mildly comfortable in one, we will spend time trashing it from the standpoint of a different discipline. This is our first one of these. And the whole point of this jump is to show on a certain level exactly where one discipline's notion of an explanation ends, another one begins. And what this one will be about is how evolution works down on a molecular level. Where did we leave off after finishing our overview of the evolution of behavior? We finished with a bunch of criticisms at the end, criticizing sort of what some of the basic tenets were of that view. First off, that notion of heritability. Heritability, assuming that all sorts of behaviors have a genetic component, have a genetic basis, have a genetic cause, and we will see those are worlds of differences in those words there. And what we saw was that that could be the case, that could not be the case. That's going to be what we focus on a great deal today. Another one of the basic tenets, this notion of adaptation. Everything you see is wonderfully adaptive. The outcome is exactly the process that evolution brings about to optimize results. The counter view being the world of spandrels. A lot of the time stuff gets carried along as baggage. Things evolving aren't necessarily things that have been sculpted into being as adaptive as possible. Another critique, and one that's also central today, is that whole emphasis on the gradualism, on small incremental changes. And what I alluded to at the end the other day is going to see a viewpoint where something very, very different is proposed to be going on, one that will make sense only eventually when we see some pretty unexpected things about genetics and the molecular biology of. Okay, so starting off, what we also finished with was a notion that there are political agendas that run through every aspect of this subject, and if that very first lecture back when talking about how some of these viewpoints influence who gets a lobotomy, who winds up exterminated, who winds up being viewed as educatable or uneducatable, etc. Political views run throughout all of this. You will see very strongly in this realm. Okay, so that viewpoint of the sociobiologist, evolutionary psychologist the other day, one of the tenets going after the notion of heritability. The notion that these behaviors are heritable or are genetically influenced, blah, blah. How would folks in that business do it? What they would do is exactly what we were seeing, which is say, okay, here's some behavioral structure, and we know how evolution works, and evolution of behavior, and individual selection, and kin selection, reciprocal altruism, and evolution of, and all the above. And then they say, well, with that framework, that explains the behavior pretty well. Go show me something better. Show me a better theoretical structure that's even more explanatory, more predictive, and until you come up with something better, I'm going to assume that this one is right, and this one comes with a large assumption of heritability of genetics, all built around inferentially. These are highly structured models built around how genes work. They explain what we see with these behaviors, and until you can give me a model that does even a better job, I'm sticking with this, and this counts as my evidence for a genetic component to this set of behaviors. And what we'll see is that's exactly where the molecular biologists decide that they finally have gotten free of people doing, I don't know, poetry for all the science in it. My God, that counts as science, coming up with a bunch of these rules there and say, until you come up with fancier rules or a more pleasing just so story, I win. That counts as science. Complete contempt for this approach. This is where a more molecular stance about it all takes off. And what we'll see that not only will have lots to say about adaptiveness, it will also have lots to say about gradualism. Okay, so starting with it, in order to make sense of this, for a molecular biologist, what is evolution about? When you see traits that have evolved, what's it about? By necessity, well, one is immediately talking about our genes. Genes, in this case, not as some constructs that one wants to maximize copies of and ones that your cousins have only whatever percentage of in common with you, but genes instead molecules, genes as information, genes as strings of DNA. And I'll assume by now there's like a basic level of shared knowledge about this after hearing that a lot of folks came to the catch-up session last week, which I think is a good idea. But what is all built about, all of this notion of genes eventually way out the other end having something to do with behavior, is the intermediary of proteins. Proteins are important. Proteins are important not just to have in your diet and make you run fast and strong and all. Proteins are in lots of ways the structurally most important things you've got making up cells. Proteins have endless roles. Proteins hold the shapes of cells together. Proteins form messengers, hormones, neurotransmitters, all of this to come. Proteins are the enzymes that do all sorts of the most important stuff. Proteins, et cetera, et cetera, et cetera. Proteins are the workhorses of what have cells doing what they're supposed to do. So the question, of course, becomes what codes for proteins? And this is where genes come in. Our basic issue here is this flow of information. Genes specify proteins. Proteins are made up of constituent building blocks, amino acids. There's approximately 20 different ones that are commonly occurring, and each one has to be coded for with a different DNA sequence. A different DNA sequence of 3 letters, 3 nucleotides. And I hope I'm not hitting a level here where people for whom this is new, this is going way too fast, and everybody else, this is way too boring, but hang on for a while in that case. Okay, so DNA codes for amino acids. A long string of DNA coding for a sequence of them, code for a sequence of amino acids which get plugged together, and you then have a protein. You got one intervening step, which for our purposes is like not really interesting and we could ignore for the most part, which is there's an intermediary step. Genes at the level of DNA, sequences of DNA, first specify an intermediate form called RNA, and it's from that that you get the readout forming the proteins. Now, everything about the function of this is built out of the following sequence. If you know the sequence of DNA, you will know the sequence of RNA. You will know the amino acid sequence. You will know the protein that thus is made. You will know the shape of the protein, and you will then know the function of the protein. And that is this critical link between what DNA, genes, evolution, blah, blah, are about, and the actual things that pop out the other end and do something. For the very critical reason that everything about protein function is built around shape. Here is a cliche that is required by law to be said at this point, which is all sorts of effector proteins fit into other molecules, other effector proteins like a, okay, everybody say it out loud. Everybody know this cliche? It fits in like a, yes, okay, cliched education at its best, like a lock and key. For those of you who are not tortured with this one from early on, it's the whole notion that a shape of a protein imparts information insofar as it interacts with something else of a shape which modifies it, which complements it, whatever. The whole world that we will have of various hormones and neurotransmitters will consist of hormones and neurotransmitters going into receptors where there's a critical relationship in the shape between the messenger and the receptor, and all of this is driven by proteins, protein shape, etc. All of this is driven thus by DNA sequences. Shape is everything with this. What you get from that is, of course, the question of where do you get different shapes from? And those 20 amino acids, for our purposes, for the purposes especially people without a strong chemistry background, all that's pertinent here is the 20 different amino acids have different degrees of being attracted to or repelled by water. which most proteins are spending their life swimming around in water. They are pulled towards water, they avoid it, they are hydrophilic, hydrophobic. All that means is different amino acids gets pulled into different positions by their relationship with water, and thus a string of amino acids, the shape it winds up getting with is determined by that amino acid sequence. There's a whole world of really interesting, horrible neurological diseases called prion diseases, which show that everything I just said is wrong, but for our purposes, everything I just said is right. So this is where you get this critical relationship: know the DNA sequence, know the coding, and out the other end, you will get protein shape, because of this business of different amino acids having different relationships with water, and thus strings of them coming up with different three-dimensional structures, and out of that will coming function out of the famed lock and key nature between shapes of functions and shapes of other things. Okay, just once again for people fairly new at this, no diagram there. One of the most interesting, or the blank slate. One of the most interesting things that proteins do is when they are enzymes. Enzymes, everybody knows that enzymes are important because they put them in your laundry detergent and advertise about how cool it is that you have enzymes in your detergent. But what enzymes do is they catalyze reactions. They cause reactions to occur, which left on their own would be very, very rare events. What enzymes do is accelerate vastly, a gazillion times over, the speed with which these happen. Catalyze reactions, what do I mean by that? For our purposes, they can take two things that aren't connected and stick them together, or they can take one thing and break it apart. For our purposes, that's what enzymes do. Virtually every enzyme out there is a protein. So proteins fit into other things and send on messages. Proteins are enzymes, pull things together, pull them apart. Proteins are structural. Again, a whole lot of the superstructure of your cells are held together by proteins. This is the realm of what they do. Notice here, suddenly there's a change of shape in a protein if it's an enzyme, because what's it doing? In some way, it's got to be pulling apart something or putting something together. The shape in some cases also can change as a function of what this protein is doing with its job. We will see a classic version of that are channels. Channels in which chemicals can flow in or out of cells, ions for chemistry types, channels that will open up under some circumstances, close under others. So protein structure not only gives shape and function, but it gives the circumstances where the shape might change in a functionally relevant way. Okay, so out of all of this comes the central dogma of life, and this was proposed by Francis Crick of Watson and Crick fame. And Francis Crick was the one who formalized saying the central dogma of how life and information flows is DNA to RNA to protein. And that became defining. An entire generation of babies were told that at birth. This is the flow of information. It has been violated in all sorts of interesting ways, which will dominate a lot of what comes. But this was the central dogma. One way in which it's violated, and again, this notion is not DNA to RNA, but the notion of whatever your DNA sequence is, whatever the gene is, whatever structure of a protein it codes for, the flow of information is going to be from DNA, RNA to protein, DNA as running everything. Note the importance of that in the very statement of this as the central dogma, the canonical flow of information in life. It all starts with DNA. DNA is the one sitting here deciding when information is going to flow from DNA to RNA to DNA as knowing what's happening. And a lot of what we will see shortly is DNA knows squat. DNA is not making a whole lot of decisions there. But one simplistic way in which central dogma went down the tubes in the 1970s or so, and one that's tangential here, but just as a first blow against central dogma, one of the things that's interesting is there are things called viruses, and what viruses do for a living is get into organisms. Viruses, in the classical form, being little smidgens of foreign DNA, which are able to get into the DNA of your own, hijack the processes there, and make the cell function for its own parasitic, vicious needs. And so you're changing the starting step of how the central dogma of life, and thus you're going to change RNA and protein, all of that. In the 70s, it became apparent there was one weirdo viral world of viruses made out of RNA. This intermediate form. All sorts of people were extremely upset about this and tried to ostracize the scientists who came up with this and told them there's no way. But eventually what was shown in Nobel Prize winning glory was there's a class of enzymes. Enzymes, they pop up, a class of enzymes that could take the RNA information and turn it back into DNA viral information, and then it does its thing. Huge blow to the central, here's information running from an RNA virus somehow being reversed back to a DNA form, and thus these are called retroviruses. Insert in the DNA, and off they go from there. So this was a major blow. Everybody eventually came to terms with this, but it still is a minor footnote in this Crickean world of everything flows from DNA. It is the Bible, it is the lawgiver, it is the Holy Grail, it is where it all starts. So stay tuned. Okay, so given that framework, one should immediately get mighty impressed with what if something changes in the DNA? What if one of the bits of coding is coded incorrectly? What if we now have on our hands a mutation? And what we will focus on here is classical realms of mutation in genetics and how that plays out in classical gradualist